Okay, in this video, we're going to talk about Criterion C1 for science lab reports in the MYP. Okay, Criterion C1 is presenting the recorded data in tables and graphs. That means that there are a number of objectives that we can break this criterion down into. The first objective is to present your raw data and your observations. So the raw data is the data that you've collected, but you haven't yet done any calculations or processing to. You need to present that data in a neatly, logically organized data table where you've labeled the data table, explained what the data is, and included relevant and appropriate units. Beneath that data table is a good place to list observations that are relevant, things that you saw. Usually it's things that you saw. It could be things that you smelled or felt or heard. These are things that you could just bullet point beneath the table as observations for the lab. Those are qualitative, whereas the raw data is quantitative and has to do with the numbers that you recorded. After that, you have to show data processing. That means carrying out at least one example, one sample calculation of each type of calculation that you did in your lab report. Third thing, take all of the processed values that you've calculated and organize them into a second data table. That data table, of course, should also be clearly labeled, have appropriate units, and be logically organized. And then finally, take the process data and present it in an appropriate graph. Depending on the exact investigation that you're doing, that could be a bar graph or it could be a scatter graph with a trend line. So I'm going to walk through an exemplar to show you what those different criteria or objectives look like. For this lab that we're looking at, this lab used a data logger. It used a data logger with Pasco Capstone software that was recording pressure over time. So the true raw data for this lab is the data that's in the Capstone program, the time data and the pressure data. So rather than submitting all of that data because it's a bit too lengthy, this student has included a screenshot that shows a sample of the raw data. This won't be relevant for all Criterion C lab reports, but if you are using a data logger, your raw data is the data that's in that Capstone software. So you should include one sample screenshot as evidence of your raw data. The raw data then gets processed. So this student has also included a screenshot that shows the processing of the data. In this lab, that involved fitting a trend line to the data and recording the slope from that trend line. So Capstone has done most of the work for the student, but this is evidence really of objective 2, showing a sample calculation, showing data processing. In some labs, there won't be Capstone software data to worry about. You will have collected the data directly yourself in class with some other sort of equipment. And so that would bring you here as your first objective. This data table here is a raw data table. It says so, it says it's raw data. And there's even a sentence after that that describes what data we're looking at. This is really helpful. Think of your lab report as being something you're going to hand off to someone who doesn't know anything about your investigation. You should be adding enough detail so that somebody who doesn't know what your investigation is about can follow it. So this has a sentence that describes what's in the data table. Then it has a logically organized data table. You can see the 3 different trials are clearly labeled. They're showing rate of reaction values, and there's a unit in parentheses next to rate of reaction. The independent variable, which is on the left here, concentration of HCl, is listed down the left-hand side. And it's in a logical order. It goes from the most concentrated down to the least concentrated. That's not the only order possible. This person could have listed it starting at 0.2, going up to 1. Instead, it could have been increasing. But either way, it makes sense that it's in order rather than to be randomly organized. So that's strand 1. They've presented a really neat, clear data table. Everything's labeled properly. The units are there. This is the raw data. Now, one thing I don't see, this person doesn't have any observations. So beneath this data table, ideally, there would also be some observations describing things that the students saw or heard about different trials in this investigation. Okay, next, this student has a process data table. This is objective 3. All process data has been organized logically into a data table. Now, this student has chosen to put this before the sample calculations. That's okay. It can also come after the sample calculations. The order is a little bit less crucial, but it's more important that you have it. So in here, they've included, again, those independent variable values, the different concentrations they tested. Then they've included all the average values that they calculated. And in this lab, they also calculated something called uncertainty. And so they've included all of the uncertainty values that they calculated here. So this kind of summarizes all of the final calculated values that will show up in the graph. Next, we're looking at the data processing. Now they've chosen to present this data in a table. You don't have to do that, but it is really well organized and it's really easy to follow this way. So this table says what calculation they're doing. So in this first row, I know that this student is calculating the average value. They've explained their method very clearly with lots of detail about how they're using Excel to do the average calculation. They've even explained how to calculate an average in general, which is even better. It's a little bit above and beyond what would be expected, but not a bad idea to show you understand what you're doing, what the calculation is. So this is how you calculate an average. You add up all the trials and divide by the number of trials. And then over here, they also have it broken down step by step. And they've included, for Excel, they've included a screenshot that shows evidence of them doing the calculation. Now, I think there's more here than is absolutely necessary. If you just said, I'm calculating the average, and then you did this column on the right, the sample calculation, where you showed the steps, you explained the steps of what you have to type into Excel, and then you show a screenshot as they've done here, that would be enough. You don't need to also explain the method and also show the equation. This is more than is fully necessary. However, you should try to set it up as clearly as this student has. So explaining what you're calculating, explaining exactly how you did it, whether on a calculator or with Excel, and then showing a screenshot with Excel as evidence that you actually did this calculation with your numbers. They've done it again for the other values that they needed to calculate in the lab. And then objective 4 is a graph. So the graph also has a brief description, a sentence that explains what the graph is about. It has a title that's very clear. It has axes that are labeled properly that have units. And for a scatter graph, it has a scatter graph, it has error bars, and it has an equation with the equation presented and the r square value presented. If you're doing a bar graph, you won't have an equation or an r squared value or a trend line, so don't worry about not having those. But the idea of presenting your data in a graph to sort of end section C1, that graph should be really clearly labeled with units and have an informative title or also a description to introduce it so that anyone looking at this graph, even if they know nothing about our science class, would be able to understand what's being presented in the graph. And if you can complete all of those objectives, you are done with Criterion C1.